What happened, Lakshmi? Is everything okay? Is everything okay?
Yes, ma'am.
Okay. So today let's start with the blotting process, and then I'll discuss about flow cytometer. Okay.
Okay, ma'am.
Okay. So let's discuss first of all blotting techniques. There are actually 4 types of blotting techniques, mainly 3 types, but another one, that is Eastern blotting, that is separate. But you just consider southern blotting, northern blotting, and western blotting. These 3 are most popular. Okay, so blotting, why we do blotting process? So through blotting process, we do the identification of what? DNA, identification of RNA, and identification of proteins. Okay, so now if you consider about the Western blot, through the Western blot, we generally identify proteins. So this is specific for protein. Southern blot means it is specific for DNA, whereas Northern blot means it is specific for RNA. Another one is there, that is Eastern blot, that is for the detection of protein after post-transcriptional modification. Okay, whether the post-transcriptional modification is happening or not, that is can be detected through Eastern blot. Okay, so Western blot, Southern blot, Northern blot, and Eastern blot. Okay, I have given one mnemonic snowdrop. I told you, Hannah. You have to remember which one is for which technique.
Yes, ma'am.
So it was a snowdrop. Snow means this one. Okay, S for DNA, N for northern blotting, and R for R. Okay, so you understood it, na? The last time I told you snowdrop. In this way, you can remember. Okay. Now, and another way you can remember that first discovery was southern blotting. Okay. So southern blotting is actually DNA identification process. So starting point is from the southern blotting, and that was done by Sir Edwin Southern. According to this scientist's name, this technique is known as the southern blotting. So DNA. So DNA is specific for southern. After that, you just remember western blotting, that is specific for protein. So other than these 2 things, you can easily recognize that northern blotting means RNA. Okay. Acha. In case of your southern blotting, what we do in that case, first of all, you have to do agarose gel electrophoresis. Okay, agarose gel electrophoresis you have to do. So after that, what you have to do? So in agarose gel electrophoresis, you will find several bands. You want to isolate one specific DNA, isolate or identify one specific DNA. In order to do that, you have to transfer these bands onto on the surface of one what is that membrane. Okay, so various types of membranes are utilized: nylon membrane, nitrocellulose membrane, most popular. So you have to transfer all the band on the surface of membrane. So it is just exactly copy of the gel. Okay, so on the membrane, what you have to do? You have to add you have to add specific probe, so that at the specific position only that probe can bind. Okay, so and after that you can identify it through the autoradiography, especially through the autoradiography. So in this process, what is done? Detection process is actually through the probe hybridization. I told you probe hybridization is, if you consider one single stranded band is there on the nitrocellulose membrane. Okay, on the single stranded band, you have to add probe, one short chain single strand, and with probe, what is attached? One radioactive compound is attached. Okay, so this is in this way, this is actually known as the hybridized DNA. Okay, we are trying to identify this DNA. Okay, in order to identify that, you have to add complementary DNA, short chain DNA, which is attached with one radioactive compounds or fluorescent compound. Okay, so in this way, you can identify your specific DNA, or you can isolate specific DNA. In order to do that, these are the steps involved during the southern blotting. If you consider, first step is the DNA isolation and degradation, and after that you have to do agarose gel electrophoresis, and third step is the DNA bands are transferred to nitrocellulose membrane. Fourth step is you have to actually denature the DNA on the transferred filter paper. Denature, you have to do denature. Why? Because you have to do the hybridization. Na? In order to do hybridization, you need single strand, not the double strand. Okay. So single strand is required. Now add probe. So fifth step is addition of probe. That means short chain DNA, single stranded DNA attached with radio labeled compound. Okay, now you just visualize it through the through X-ray film, or you can do the autoradiography technique. Okay, this is the case what we have done during the southern blotting process. Okay, in this case, how bands are transferred from gel to nitrocellulose paper. This is the arrangement. Okay, so first of all, in a tray, buffer is given, and first of all, gel is kept. After that, you just add membrane. Okay, you just put one similar sized membrane filter paper. Membrane means here mainly nitrocellulose filter paper, nylon paper. Now you just add paper, filter paper, then paper tissue, then glass plate, and after that you add some weight, so that this arrangement will facilitate the transfer of all the bands from agarose gel to membrane. So in this case, we are doing attachment of membrane and gel. है ना? Then only transfer is possible. Okay, and in this case, what is happening, through the capillary action, from the buffer is going towards the gel, and from the gel all the bands are transferred to the nitrocellulose membrane, so by capillary action. Okay, so that is done. Now in this case, same thing is given in a different way. New thing, one step is involved here, that is the restriction fragments present in the gel are denatured. So alkali treatment is given here. So alkali treatment is actually done on the gel, and then it is transferred to nitrocellulose membrane. Okay, it's transferred onto a nitrocellulose filter or nylon membrane. Is it clear? Can you tell me what are the steps involved? If you want to watch this diagram, you can follow that. Otherwise, you can tell me your own word. You tell me. Try to explain what are the steps involved in the Western blotting process. Sorry, in the we are talking about the southern blotting. Lakshmi, tell me. Is it possible for you?
Yes, ma'am.
Hmm,
First,
try to try to.
DNA digestion. We'll do DNA digestion using restriction enzyme.
Hmm.
Then restriction enzyme will cut the DNA at specific sites, and it will generate fragments of DNA.
Hmm.
And those fragments we will allow it to run in gel electrophoresis.
Hmm.
Uh, After that, it will be we will let that to denature because
Very good. Huh.
single strands.
Hmm. Denatured by alkali treatment. Okay, NaOH. NaOH is the alkali, so you can add alkali. Okay, now then
and then we'll transfer into a nitrocellulose membrane.
Mhm. Now you have to add the
Oh,
probe,
mm,
radio labeled probe,
mm,
and finally you allow them to bind. Hybridization, you have to allow for the hybridization process, and after that you can visualize it. You wash it and visualize it. है ना? So
mm.
unbound everything will be removed. You can easily identify the band of interest or DNA of interest. Okay, so this is all about southern blotting. So southern blotting can be utilized whenever any process where DNA, you have to identify the DNA. So in that process you use southern blotting process. Okay, so in the gene discovery, mapping, evolution, development, studies, diagnostic studies, forensic science, everywhere you can use this type of method in order to identify the DNA. Now next topic is the northern blotting. Northern blotting, it is actually developed by James Alwyn and George Stark. So here is no similarity with the name of the technique, with the scientist's name. Okay, and northern blotting, but developed by different scientists, James Alwyn and George Stark. Okay, why it is given northern blotting? In order to, you can give the you want to give the similar name like southern blotting, it is given northern. Okay, nothing else. There is no relation with any scientist here. Okay, so southern blotting actually developed first. After that, this northern blotting. Okay, southern blotting, it was developed in 1975, and this is in 1979. Now in northern blotting, what is the steps involved? What are the steps involved? Exactly same like DNA. Exactly same like DNA. Difference क्या है? You have to run RNA. Okay, you have to run RNA. One difference. Another is most of the cases amino benzoyl oxymethyl filter paper is used. Nitrocellulose nylon is also used, but this is mainly utilized for the RNA transfer from the gel. Okay, so here every in this slide, na DNA is given. It's not DNA, it's RNA. Okay, this is RNA. And okay, now see, all the steps are similar, exactly similar. See, RNA is clipped, electrophoresis is used to separate the RNA. Then RNA fragments are blotted onto amino benzoyl oxymethyl filter. Okay, so that is happening. You just chalo. Uh, your friend, I think,
Yeah, I sent her the link of the
Shraddha.
app. Mm.
Is it possible to whether she has the account for Plus Plus?
I think
Don't
we
know.
just need a phone number. I don't know. I just sent her the link to log in. She might try.
Okay. Okay, so just similar. Northern blotting is similar to southern blotting. And here also same thing is given. So here denaturation is not required because RNA is itself single strand. है ना? अच्छा. Samples are loaded on gel and the RNA samples are separated according to their size on the agar gel. Now the gel is then blotted on a nylon membrane or amino benzoyl oxymethyl filter paper by creating the sandwich arrangement. Then the membrane is placed in a dish containing hybridization buffer with a labeled probe. Okay, labeled DNA probe here, single stranded DNA, I think, binding with this RNA. And then the membrane is washed to remove unbound probe. A labeled probe is detected via autoradiography or via a chemiluminescence reaction. Okay, and in both cases, this results in the formation of a dark band in the I eat one chocolate biscuit all in one band on an X-ray film. Okay, so this is all about the northern blotting. Now application. So whenever you want to identify the mRNA, you have to use it. So detect the expression level. If you want to detect the expression level, so you have to measure mRNA. You can identify the mRNA through northern blotting. Okay. So detect the expression level and transcript size of the specific gene in a specific tissue or at a specific time. Sometimes mutations do not affect coding regions but transcriptional regulatory sequences. Okay, so mainly you just check the expression level and transcript size. Transcript size means if you consider this is the size of the DNA gene, okay. So, but whenever you are getting mRNA, size of the transcript will be less, na? So you can determine the size. Acha. We don't have to understand this thing. Not the blotting disadvantage. The main disadvantage you have to use the radioactive thing. That is for all the blotting processes. Mostly you have to use the radioactive. That is not good for health. Okay, and in this case standard relatively less sensitive than reverse archer. Now detection with multiple probe is problem because in this case we are only applying one probe na? So if you use one probe, detection is better. One probe, you just trying to identify one band. In that case this is very useful, but if you want to identify multiple bands, so it will be problematic. Multiple sample cannot be identified in a single run. Okay, so that is problem. Now, if RNA samples are even slightly degraded by RNAs, the quality of the data and quantitation of expression is by negatively affected. So these are the disadvantages, nothing else, nothing important. Now, Western blotting. Western blotting means you have to detect the protein. So in this case, mainly we do check the protein expression levels. Okay. So it is, in other words, Western blotting is used to determine the protein expression, and you can determine the size of the protein, amount of the protein. Okay. So these are detected through the Western blotting technique. So this figure is given for the Western blotting technique. It's just like indirect method of ELISA, where first of all you have to add the antigen. Now add primary antibody for your antigen of interest, है ना? Then you just add secondary antibody. Secondary antibody is actually attached with any enzyme or any additive compound. Okay, so this is actually about the indirect method of Western blotting. If you want to do the direct method, you just add the probe, or not probe, you just add the label of enzyme or radioactive compound with primary antibody. That will be direct. So antigen and primary antibody will be there only. There will be no secondary antibody in case of direct method. Okay. So what are the steps? Exactly same. Sample preparation, separate proteins, transfer to membrane. Separate protein through which method? Which method? Separation of proteins?
Western.
No, separation of protein here. First of all, you have to do the SDS page. So in case of DNA and RNA, always remember, in case of DNA, RNA, we are using
Gel electrophoresis.
gel electrophoresis, not like but the agarose gel electrophoresis specifically. Most of the cases we use agarose gel electrophoresis. Whereas in case of protein, we generally use SDS page electrophoresis. Okay, polyacrylamide gel electrophoresis. That is the page, SDS page.
Yes.
Okay, why? Because agarose gel generally, agarose porosity is higher than, porosity is more than this polyacrylamide gel.
Mm.
Okay, so DNA RNA generally thread like structure, na? The size of the DNA RNA will be more bigger. So agarose, through the agarose gel separation will be better. They can move faster through the agarose gel. But protein is generally, most of the cases you will see globular structure. है ना? So if porosity is more, acrylamide gel means this type of mesh structure will be there. Okay, mesh will be there. So small porosity is there, but protein can move through this small porosity. Okay, so therefore SDS page is most of the cases we use for the protein separation. So this is the difference. We are using SDS page here. In case of DNA RNA, we are using agarogel electrophoresis. Now next step is same. Just transfer the protein to membrane. And now new step is involved here. That is blocking of the membrane. Why blocking of the membrane? Here now we have to add the primary antibody on the surface of membrane. That is the next step. But antibody can bind with the membrane. Okay, so inside the membrane you have few bands specific for the protein, but vacant space is there na, on the paper. So
Mm.
at the vacant space, actually primary antibody can bind on the paper. There will be non-specific binding. Therefore, in order to block that non-specific binding, we are using what? We are using blocking agent, that is mainly BSA or milk. है ना? Milk is also protein, milk protein. So they will bind with the filter paper. So there will be, everything will be covered by the milk or BSA. Only the protein bands will be independent. Now, if you add the primary antibody on the surface of this membrane, antibody will bind with only specific antigen what you want to identify. Okay, now if you wash it, what will happen? Unbound samples will be removed. If there is specific antibodies present in the plane, you will get band. Okay, okay, so finally you can detect it. So this is the case in case of western blotting. So exactly everything is same. You can, basic method, basic principle same, है ना? Slight side difference is there, nothing else. If you know the property of DNA, protein, and RNA, you can easily identify the difference. What are the differences? Okay, so in this case, you just follow these steps: preparation of sample, protein extraction, SDS page, protein transfer, blocking the membrane, then detection. So these are the main processes involved. Acha, in this case, nitrocellulose membrane is used as well as PVDF membrane is used. Polyvinyl difluoride something is there. PVDF membrane is used. So this membrane is mostly utilized for the protein separation. Okay, protein transfer. Clear? So here milk or BSA is used as the blocking agent. So this is also new here. Achha. Application everywhere. If you want to identify any antigen, viral, bacterial, you can use this. So this application is almost exactly like ELISA application, है ना? ELISA application. Okay, so here see HIV detection, HPV detection, HAVS detection, and BSC detection, bovine spongiform encephalopathy, then Lyme disease. These are all by any parasite or any virus or bacteria. So you can identify those molecules as the antigen. So through Western blot, we are determining protein. That means you can identify the antigen, and antigens are mostly mostly proteins. Acha. In case of SDA space, generally we use vertical gel electrophoresis unit, whereas in case of agarose gel, we generally use horizontal gel electrophoresis. So Western blot is actually done in vertical electrophoresis, whereas southern blot and northern blot, those are actually in that case horizontal agarose gel electrophoresis is done. Okay, so these all three methods involved. Now going to Eastern blot. In Eastern blot, you don't have to understand anything. You just understand that in this process, we generally determine the whether post-translational modification is happening or not. Now, what is post-translational modification? I have given a lot of examples during the discussion of translation process. Okay. One example was phosphorylation and carbohydrate addition, that means glycosylation. Glycosylation is the main post-translational modification you can consider, because most of the cases after the translation, all the proteins should be transferred to different organs, na? So that transfer is done through the glycosylation process. Whenever glycosylation is done, then only that they can be transferred to different places. Okay, so you can determine that whether carbohydrate moieties are present or not. You can determine through Eastern blotting process. Okay, here it is written Eastern blotting is a biochemical technique used to analyze protein post-translational modification or PTM, such as so carbohydrate may be added, lipids can be added, phosphorus moieties can be added, or carbohydrate moieties can be added after the translation process. Now it is most often used to detect carbohydrate epitopes. So most I told you na कि most popular post-translational modification is the addition of carbohydrate moiety or glycosylation. है ना? Addition of carbohydrate moiety is known as the glycosylation. Okay, so detect carbohydrate epitopes. So region where carbohydrate moieties are attached, in order to identify that, you just follow Eastern blot. Thus, Eastern blotting can be considered an extension of the biochemical technique of Western blot. It's almost like a Western blot, but it is specific for the carbohydrate. Sorry, for the carbohydrate moiety. Okay. Achha. So in this table, what is given? All the mainly three methods are given here: southern blotting, northern blotting, western blotting. So in this case, uh, in southern blotting, we are detecting DNA. In northern blotting, mRNA. In western blotting, we are detecting protein. Gel electrophoresis, gel electrophoresis utilized in southern blotting, it is agarose. In case of northern blotting, it is agarose or formaldehyde agarose. The now western blotting, polyacrylamide gel. Gel pre-treatment may be done in case of DNA for the denaturation. Okay, and other cases there is no pre-treatment. Now blotting method, capillary transfer. I told you na, capillary transfer means in a container, first of all you have to put buffer. Okay, so buffer should be there. Buffer is transferred to the gel through the capillary action. Okay, through the capillary action, this buffer is going towards the gel. Now from gel, what is up above the gel, what is present? From gel, bands are transferred to which thing? What, to which place? Huh? Tell me. From gel, on the surface of gel, what is present? Membrane. Membrane is present, na? So from gel, bands are transferred to membrane. Okay. and that is through the capillary action. So capillary transfer both in case of northern and southern blot, but most of the cases in western blot we generally apply electric field. So where if you add this, what is that?
Ma'am,
Uh,
capillary transfer, how does it take place?
Capillary action means normally if you just a minute. Capillary action, how can I explain capillary action? Whenever you are using दिया जलाते हो ना, दिया बत्ती जलाते हो ना, इन
Mm,
है ना, you are putting oil in a container, है ना,
mm,
है ना, and on the surface of that you are adding one cotton thread, right?
mm, mm,
Whenever this light is on, what is happening actually? What is happening? This oil is transferred to the cotton.
mm,
This process is actually through the this process is actually known as the capillary through the capillary means capillary means, yeah, this type of uh, path is there. You can consider if one is going, so simultaneously another is also going side by side, not side by side, just below the one by one it is transferred. Or any other example, how can I give you? Now if you just make one cotton thread and put it into any water, what will happen? The water will transfer from below the place to your hand, है ना?
mm,
That is actually known as the capillary action. Now if you think this is the sponge, okay, you are putting it on a water, so what will happen? Initially this sponge will be water will be transferred to first part of the sponge, and simultaneously it will go upward motion, है ना? So
mm,
that this process is capillary, that means it is making one several capillaries. Through capillaries it is going one by one all the droplets. Okay, so it is opposite to your opposite to your, what is that, earth attraction, that is what is known as that.
gravity.
Against gravity. Okay.
Mm,
Okay, so it is making one different capillaries. Through capillaries they are moving, water droplets are moving one by one. Okay.
mm,
Acha. Now in electric field means you are applying electric field, then only all the proteins are transferred to the nitrocellulose membrane. In that case, most of the cases what is happening now, in that case, in the middle of the middle of the position gel is kept. Okay, gel
mm,
is kept, then side by side what is given, side by side actually sandwich position will be there. So here one nitrocellulose paper, okay, and here also another nitrocellulose paper. Then electric field is applied. Electric field is applied for. So what will happen? Few protein molecule will be there which are positively charged. Few proteins will be there with negatively charged. है ना? So what will happen? Few will go towards the anode and few will go towards the cathode. Okay, in this way some separation better if in the mixture several bands are there in the gel, you can separate very resolution will be better. Okay, what I am trying to say, if you consider in a gel, okay, you just trying to explain it. So in a gel several bands are there, okay, several bands are there. Now, whenever you are transferring it to nitrocellulose paper through the electric transfer, what will happen? In this protein, few will be positively charged. Okay, if you consider this is positively charged, these two are positively charged. Okay. Now, if you consider other two negatively charged compounds are also there. These are green color is the negatively charged. Okay, green color I am again giving you separate bands. for the negatively charged. Okay. Now you just apply electric field and transfer all the bands towards the nitrocellulose paper. Two nitrocellulose papers are involved here. Okay, side by side. So what will happen? One nitrocellulose paper, if you consider these are green color is the negative one, they will go towards the anode. Okay, so which nitrocellulose membrane is present towards the anode? So these green colored band will go that position. So you will get only two bands in the nitrocellulose membrane if you are putting it towards the anode. Now, if you consider that you are giving right part is the cathode. Right part nitrocellulose membrane is present at the cathode position. Cathode means it is negatively charged. So all the positively charged molecules present in the band, they will go towards this nitrocellulose membrane. So you can separate. You will get 1, 2, 3, 4, 5, 6. Total 6 bands you can get here. 6 band, not two positively charged is there. So you can get only 2 bands. Clear? If it is neutral, they will not go anywhere. But other red color, if you all the red colors are positively charged, they will go towards the anode. Clear? So here separation is better, na? Initially a lot of total 9 bands were there. But whenever you are transferred to two separate nitrocellulose paper, so band separation is better. 2 here and another 7 here. है ना?
Mm.
And separately you can add antibodies against your protein of interest. Okay. Separately you just add on this nitrocellulose membrane as well as on this nitrocellulose membrane. Okay. So you can find anywhere your protein of interest. Now if you know ki after giving the your protein of interest is negatively charged, so you just follow the negatively charged, you just add the probe on the surface of negatively charged that nitrocellulose. Yeah, in this way you can identify it. Acha. Now these are the simple steps. Sample preparation. Now gel electrophoresis, transfer of blotting, probing, detection, imaging, and analysis. Finally, you can analyze it. What is happening? Always analysis is very not only experiment is important. Experiment result analysis is very tough. That is the main you can consider. You can get a lot of results. If you are not analyzing it properly, there is no meaning of that result. है ना? That we do during the research, during the PhD. So science you can consider experiment, gathering on the experimental data as well as analyzing of the experimental data. Okay, so all these about southern blotting, northern blotting, western blotting, and eastern blotting. Is it clear? So
Yes,
should I
ma'am.
go further? That Shraddha could not join, I think. After a long time, she was trying to join, but Google Meet was so good. Achha, I'm crossing this part, okay? Now
Okay,
going to
ma'am.
next topic. Next topic that is flow cytometer. So that is slightly complicated. You have to be focused, okay? Now see what is happening here. Flow cytometer. We're talking about flow cytometer. So I have taken all the data from these two sources, okay? Now, what is the main principle here in the flow cytometer? So this is also technique in order to identify any molecule through light scattering. Okay, through light scattering. So what is happening here? flow cytometry is a widely used laser based method. So in this case laser beam is given. So laser beam light is given. So laser based method for analyzing the expression of cell surface and intracellular molecules. So in that case, mostly we are determining protein. So it may be either present on the surface, okay, on the surface it is present, or it is present inside the cell. You can identify both the cases from through the flow cytometry. Okay, so internal molecule or outside of the molecule, cell surface molecule, you can identify. Okay, so now see, this is usually based on measuring fluorescent intensity. So in this case, we generally measure fluorescence intensity produced by fluorescent labeled antibodies specific to proteins on or inside the cell, or ligands that bind to specific cell associated molecules like propydium iodine binding to DNA. So here, whatever, a lot of things are given, you don't have to focus on that. You just understand that. If you consider you want to identify the cell surface protein, okay, so you just consider this is the antigen cell, one cell. If you consider bacteria cell or whatever viral cell, on the surface of cell there should be some epitope, है ना? Different epitopes may be there. If you are if you want to focus on one epitope, you just add antibody against that epitope, and that antibody will be labeled with fluorescent compound. Okay, and now you just detect the level of fluorescent light, how much it is emitting, or how much normal laser light is scattered. You have to detect through the machine, through a cytometer. Okay, so in this case what is happening, a same that antigen antibody interaction we are studying here, but generally we are adding fluorophore or fluorescent molecule which is attached with the antibody. Okay, and we are trying to measure in this through this process we can measure different size of the molecule. Okay, that means there will be some different data or different plot for smaller molecule as compared to the bigger molecule. Okay, as well as we are, so that means we are measuring size of the molecule. You can determine the size of the molecule, as well as you can determine the if inside the cell something is present, you can also determine that. That means most of the cases we check the granular molecules are present inside the cell or not. That is also determined. So size of the cell as well as any granular samples are present inside the cell or not, that is also can be detected through flow cytometer. Okay. So now in this case, acha. So what is the actual, what is happening? I am showing it. Shortcut way I am trying to draw this method. What is happening actually? So first of all, I am making one that is called fluidic, fluidic system. So there will be one fluidic system in this way. This type of fluid, it's like column. Okay, so fluidic system will be there. Okay, so in the fluidic system, a lot you just add the sample. So sample means you are adding mixture of antigen. You want to identify the one of your interest, है ना? So if there are a mixture of antigens, okay, I'm writing a lot of antigens here. So here a lot of mixture of antigens are present. You want to isolate only one. Okay. So in that case, I'm trying to give only green color. Okay. You want to isolate the green color. Okay. Now you have to add antibody against this green color. Okay, As I told you, antibody should be added, and that antibody is actually attached with what? One fluorescent compound. So it is labeled. Okay. Now you just pass this sample through this fluidic system. Now through the fluidic system, what will happen? One by one droplet will come out. One by one cell, single cells will come out. All the single cell will come out one by one. So in this way, system is designed in this way. It one only one cell will come at a time. Okay. Now all the cells are coming one by one. Now what will happen? What will happen? We have to add some laser light here. So through the laser light we are detecting it. So here should be some laser light source. So light will give, if you see, it will, this light is going, and mainly the fluorescent compound, that is green colored compound attached with the antibody, it will heat it. Okay. And after heating, what will happen? There will be light scattering, because whenever light is passing through any medium, if any barrier is coming, so if you see, this is the one particle is there, whenever any light is coming, and whenever it is on the surface of this molecule, what will happen? This light will be scattered, and it will give you scattered light, one straight scattered light, that is forward scattered light, and another light that is known as the side light scattered. So there will be one straight scattered light, another will be for the scatter one. In this way you will get a lot of scattered light. And for these two types of light in this system, there is detection process. Okay, detector will be there for forward light as well as for the side light. Okay, so there will be two types of detector, and detector will be attached with finally analyzer. Okay, and you can get how much scattering is happening here, how much FSC is scattered, how much SSC is scattered. So depending upon the result, you can analyze it. Key size of, according to the size you will get different signal, and according to the granularity, that means how much granules are present, according to that you will get SSC. Okay, side light, and you can analyze that. Okay, so this is the main thing. You are putting one laser beam, and samples are loaded inside what? Fluidic system. And in the fluidic system, samples will be coming out of the fluidic system as the single cell. On the cell, um, laser light is actually scattered, and detection will be there. Okay, so this is the main principle here. Mainly we are detecting two things, two scattered light, mainly two scattered light. Other than that, fluorescent light also detected, because fluorescent compound is there attached with the molecule. So fluorescent compound is also detected here. Okay, now going to the how how samples are prepared for the fluorocytometer. So in this case, you just take the blood. Okay, blood sample. Now in the blood cells, separate the cells. You have to get all the separate cell. See, the clump will not be helpful for this process. The individual cell will be there. You just separate it, and now add antibodies which is attached with the fluorescent compound. Okay, now antibodies will bind with the antigen. Okay, and next step is you just add this compound or mixture of the antigen and antibody in onto the flow cytometer, that is called the this chamber. This chamber is known as the, what I told you, this chamber is known as fluidic system. Fluidic system. Okay, fluids are there, so it is known as fluidic system. Okay, other than that place, nowhere is fluidic system. Only one place is there, which is known as the fluidic system. Fluids are there because you are loading the sample in that place. Okay, so in the next step, what are the actual steps involved here? Biological sample preparation. Now label it with a fluorescent marker. Then cells move in a linear stream. As I told you, key in the flow cytometer, linear stream will be there and single cell should be there. Cells move in a linear stream through a focused light source. Light source is the laser beam. Okay. And now fluorescent molecule gets activated and emits light that is filtered and detected by sensitive light detectors. So in this case, mainly three types of detectors are there. I will discuss about that. Now conversion of analog fluorescent signals to digital signals. So finally there will be in the computer you will find the result, all the plots you can get. Okay, so that is called the digital signal. Okay. Now, in this case, what is happening? In this case, using a flow cytometric machine, cells or other particles suspended in a liquid stream. Okay. Now, in this case, sample should be in the liquid form, and they are passed through a laser light beam in single file fashion. An interaction with the light is measured by an electronic detection apparatus as light scatter and fluorescent intensity. Light scatter is also we are determining. Light scatter, two types of light scatter: side light scatter as well as forward light scatter. Okay, and so these two light scatter detection plus fluorescent fluorescence intensity detection is done here. Okay, now if you consider what are the systems present here actually, in that case, here, uh, in this case, here central channel is there. First of all, central channel is there. Okay, inside the central channel, you have to add the sample. Okay, now another outside of the central channel, what is present? Outer sheath. In this case, this is the outer sheath. Okay, this part is the outer sheath. So outer sheath, it contains faster flowing fluid. Okay, faster flowing fluid, mainly saline, phosphate buffer. Okay, phosphate buffer saline. So in this case, in the outer sheath, phosphate buffer saline is given, and middle of the core, central channel, what is present? Sample present, liquid in the liquid medium. Okay, now such a system is created that samples, all the cells will run one by one. Only single particles will come out of the machine. Okay. So here, what is written here? One of the fundamentals of flow cytometer is the ability to measure the properties of individual particles. So individual, we are because laser beam is focusing only on the one particle. Okay. So we measure the scattering light uh, depending upon one particle scattering. Okay, now individual particles, see, measure the properties of individual particles which is managed by the fluidic system. Now when a sample is injected into the flow cytometer, it is ordered into a stream of single particles. This is the main thing I'm trying to explain you. Stream of single particles is very important. Now the fluidic system consists of a flow cell. So flow cell, I was talking about flow system, that is fluidic system or flow cell. So flow cell is containing actually central channel plus outer sheath. Okay. So these are two things present in that case, liquid system. Acha. Now flow cytometer is a tool because this is very powerful tool because it allows simultaneous multi-parametric analysis of physical as well as chemical characteristic up to thousands of particles per second. So efficiency is very high because within a second you can analyze a lot of data from lot of particles. Okay, so therefore it is known as the powerful tool. Within a minute, you can analyze thousands of particles within a second. Now, this makes it a rapid and quantitative method for analysis and purification of cells in suspension. Now, using flow, we can determine the phenotype. Phenotype means mainly we determine the size of the molecule and the function and even short life cells. Even why? Because this is very within a second you can determine na. So if any molecule or any cell is short life, so in that case you can determine it through flow cytometry method. Okay. Now in this case fluorescence activating cell sorting is done through this method. In that case it is full form is FACS. Okay, this is an abbreviation of this one, fluorescence activated cell sorting. In this case, by utilizing highly specific antibodies, so in this case, same whatever I told you, antibodies are labeled with fluorescent conjugates. Okay. Now, FSC analysis allows us to simultaneously collect data, sort a biological sample by a nearly limitless number of different parameters. Or nothing else. Same thing, whatever I told you, same thing. We can sort all the cells. Sorting means you just separate all the cells through this process. Now, in this case, what are the fluorophores added? So fluorophores examples are FITC, PrEP-CP, APC, PECY 5.5. These are the name of few fluorophores which are actually labeled with antibodies. Okay, so they are actually giving the fluorescent color. Okay, so in this case, if you see na, a mixture of antigens are there. Now antibodies are given. Okay, and antibodies are binding with the specific antigen, and it should be coming out with the antigen. In this case it is not given. Why it is not given? Acha, this is fluorescent dye. Antibody is attached with the fluorescent dye. This is antibody. Now against antibody, why it is given green color? Again green color. It should not be green color, it should be some different na. If this is the particle, if this is the particle or a cell, so in that case on the surface of cell there should be some antigen, है ना? There should be some antigen. So I think they have given wrongly. This figure is slightly wrong, I think, because the structure should be like one antigen is there on the surface of antigen, epitope is there. Against that epitope you are adding antibodies. Okay, and with antibodies what is present? One label, fluorophore is attached, है ना? That should be the process. Okay, acha, oval shape is given, I think oval shape is given. But in this figure, antibodies, antigen binding is not happening. So green color should bind here, na? At the AB region here, it should bind here, है ना? It should bind here. If antigen are oval shaped, so it should bind here. Now here there should be, I think in this molecule also there should be antibody attached. Acha. Now laser is actually giving the exciting the molecule, and scattering is done. So now two types of scattering I told you, FSC, forward and side scattering. So different detectors are there for the forward one as well as for the side one. Now forward one is actually for size, depending upon the size of the cell it will give you the peak, whereas side by side detector, those are actually giving you fluorescent peaks as well as for the granularity. According, if more granules are present, scattering will be more, है ना? If you consider this is one molecule, inside the molecule several particles are there, granules are there, what will happen? Light will scatter more. So in that case it will scatter not forward position, it will be scattered in the side by side position. Okay, the light scattering will be side by side position. So more scattering means more SSC means more granular. Okay, now here it is given ki such an arrangement is done ki one by one molecule or one by one particle is coming out of this system. Okay, now In this process also same thing is given, nothing else is there. Let's see any new thing is given or not. Anything new is it there? Cell suspension, ultrasonic nozzle vibrator forms the droplets. Okay, now laser light is going on one by one particle. Now detector, two types of detectors are there. And finally you can collect sample also. Okay, if you add the positive and negative electric field, what will happen, you will collect your sample. If you want to make your protein of interest negatively charged, you can make it and separate accordingly. Okay, see here, small groups of drops negatively charged due to detection of single fluorescent cell. Now, small groups of drops positively charged due to detection of single non-fluorescent cell. Now, you just apply the electric charge. What will happen? Positively charged will go one position, negatively charged will go another position, and a neutral one will go another position. You will collect those samples. So, cell collector. So, flask for undeflected droplets. So, whatever no scattering is happening, in that case, you can collect those sample also. That means labeling is not done. Okay, in this way you can collect your sample too. Not only detection is done, you can collect the sample or you can separate the sample. Okay, now this is clearly shown that there are mainly three what you can see, three main parts are there, or three main systems are there, core systems are there. One is fluidics, second is optics, and third one is electronics. Fluidic system, whatever we discussed now, two systems are there na in the fluidic system, core one and seed one, है ना? Those are coming under the fluidic system. So here, see, fluidic system includes a flow cell where the sample fluid is injected, and the flow cell requires seed fluid. So orange color is the seed, and this blue color part is the core system, sample fluid. Okay, flow cell, core or flow cell you can consider, where sample is loaded. Now second part is the optic system. Several filters are added here, optic system. So along with that you have to add here laser beam light. This is also present in this flow cytometer. Okay, laser beam here it is not added, they have only added the instruments parts. So fluidics, optics, and electronics. Okay. Now, optic system means optic system consists of various filters, light detectors, light source. Now, which is usually a laser line. Acha. So in this case, optics, in the optic system they have added the laser one also. So laser light plus different light filters plus detectors. Those are together you can consider as the optic system. Now, what is electronic system? Electronic system means data compilation is done in the electronic system. Okay. So actually detectors are attached with the electronic system. Okay. So in this way, fluidics are attached with optic system, and optic systems are attached with the electronic system. Okay, in this way, mainly three systems are there: fluidics, optics, and electronics. Okay, so from this method, you can properly understand. This is very good figure. You just add the sample, laser beam. Now, what will happen? There will be two scattering, and the SSC plus FSC, and you just detect it, and finally, you just compile the data. Okay. Acha, in this case, this is where the particles are passed through at least one laser beam. Lasers are available at different wavelengths. Acha, different wavelengths are available, and for the different fluorophore, wavelengths are different. Okay. Now, lasers is available at different wavelengths ranging from ultraviolet to far red and have a variable range of power levels as well. Interrogation by the laser beam excites any compatible fluorescent probes that are conjugated to antibodies, causing the probes to emit light at specific wavelength. Okay, for the chromophore, depending upon the chromophore, uh, wavelength of the light will be different. Okay. Now see in detail about the forward scatter light signal and side by side scatter light signal. See, key parameters which are measured by this machine include forward light scatter, side light scatter, as well as fluorescence emission signal. So three things are involved here. During the detection process, you have to check these three things. Okay, so forward means depending upon the size, you will get different scattering light. That means if you consider key particle size is like this. Okay, now light is coming, laser beam is coming. So after the scattering, you will get this type of scattering, forward scattering. Okay, up to this. Now, whenever the size of the particle is increased, okay, so whenever it is increased, suppose this is the, okay, just a minute. I'm increasing the size. If you increase the size of the particle, what will happen? Scattering will be more or less. Whenever you are at just this is, suppose this is laser beam. Now after the scattering, forward scattering will be much more scattering will be there. So this will be like this. So whenever particle size is increase, so forward scatter light signal will increase. Clear? In this way, if increase amount of forward light scattering if you find, that means you have to identify that size of that particle is more than previous one. Okay, in this way you have to do nothing else. Now if you consider next thing, side by side. Side by side, what I told you, side light is actually we determine the granularity, how much amount of granules are present inside the cell, we detect through the side scatter light signal. Okay, and as well as complexity inside the cells, how much amount of other compounds are present. So that is granularity is also coming under complexity, you can consider. Okay, so how much complex form is there? If more complex, more granular form is there, that means more side scatter light. Okay, so now cells with a low granularity and complexity will produce less side scatter light, while highly granular cells with a high degree of internal complexity will result in a higher side scatter signal. This is the main thing in case of side scatter light. Clear? Okay. Hmm? So here in this case, side scatter not, we are not measuring the size of the molecule, size of the particle. Although due to the size of the molecule it is affected, but it is more sensitive towards the granularity, complexity. Okay, and it is also sensitive to membrane, cytoplasm, nucleus. That is also there. Acha. Now, nothing important from this now. Uh, so I told you three detection is required. Na? Three types of detection: forward light, then side scatter light, and third one is fluorescence emission signal. I told you that thing. Acha, in this case, is it given fluorescent? No, in this figure it is not given. Only forward and backward, forward and side by side scattering is given. Fluorescent वाला नहीं दिया है. In the next picture I will give you how it is happening. So third one is detection of fluorescence. So fluorescent light emission signal. So in this case, if you are identifying antigen, you have added antibody and you have added fluorescent compound with the antibody. We have to detect that, nothing else. Here, one thing is to note that fluorescent signals may also occur from naturally fluorescent substance. Acha. Sometimes you will get without addition of any fluorescent compound, these molecules can give you fluorescence. Okay, NADPH and FAD, they will give you fluorescence peak. Now, in general, larger and more granular cells have higher levels of autofluorescence due to having an increased amount of fluorescent compounds. So, in this case, therefore, na, in my previous slide, I think I told you, he, where I have given and one picture was there. Yes, in this case, I have given a side by side scattering means it is giving you granularity result as well as fluorescence result, है ना? So that same thing is given here only. See, it is given in general larger and more granular. If more granular, then also you will get fluorescent light more, emission more. So cells have higher levels of autofluorescence due to having an increased amount of fluorescent compounds. Okay, so this is happening now in this picture. Together side scattering as well as forward scattering is given. So green color is the laser beam. Now this part is forward one, or few other this thing also you can include. Okay. Now side scattering means these are actually side scattering. These are side scattering. Okay. Light. Now see, all cells or particles passing through the beam scatter laser light measures as forward scatter and side scatter. Now you can plot it y versus x. If you want to plot in the x axis, if you measure side by side scattering, you can plot in the y axis as the forward scattering. Okay. Now if S correlates with cell size, SS is the proportional to granularity and cell. Same thing several times it's written, nothing else. Okay, in this way, for one particle, you will find one peaks. Okay, one dot result. How it is coming out? Let's see. This is the picture actually. You will see a lot of dot. For every particle, you will get peak. Okay, so if you consider this is the x-axis and this is the y-axis. So this is actually giving you the forward one. No, here, so it will measure the
And the
forward
video
and the
is
side.
freezed.
Huh?
The video is freezed.
Again,
Which
I
slide
don't.
you are in?
I am teaching you 16.
It's freezing 15.
Yeah,
One
wait for two
second.
minutes. Wait for few minutes. Just 15 is visible. I'm writing something on 15. Is it visible? Green color? No,
No.
no, not coming. You just rejoin. Rejoin.
Ma'am, I'll leave and join again.
Ha. Therefore, I'm telling you, rejoin. Okay, now is it visible? Now this is 16 slide. Okay, so you will get this type of result. Okay, so this is for single particle you will get the plot. Okay, now according to this, from the data you can analyze. How you will analyze, I will show you in the separate slide. You just understand this. In the x-axis you can plot, your size, no? So in this case, in the x-axis, you can plot what? You can plot forward angle light. I told you opposite one. In x-axis, actually, you have generally you can plot Fs, and in y-axis you plot which one? Side by one. And as SS. So what from the plot you can understand, if monocyte peak is here, you just divide this into four sections. What will happen? If monocyte is coming here, that means
Ma'am, it
if
freezed
S
again.
again freezed.
Mm.
If I
Ma'am,
count,
my drawer and network.
from your side problem is there. I think here no problem. This is very important topic. I have to complete it. Wait for a few minutes first. Hello.
Yes, ma'am.
So no, if S value is higher, that means if you consider this is the monocyte, na right side part is monocyte is given here in this position,
Mm,
है ना? So
mm.
monocyte is given. So this value is showing that Y value is less, but X value is high, right? This monocyte, if you consider
Hey,
the
I was
monocyte,
about to wash. I was about to wash after class. Uh,
hello.
yes, ma'am.
So if you consider the monocytes, in this case, X value higher है, but Y value less, right? So
Mm,
X value higher means Fs value higher. Fs value higher means size of this molecule is higher, right? According to the size, if size is more larger, so in that case, Fs value will be higher, as I told you, है ना? Monocyte is showing कि X value, if you this is the X value na, X value higher
mm,
position as compared to this debris,
mm,
है ना? Now if you consider this part is showing that if his value is high as well as S value is also high. S value is showing what thing? Granularity.
mm.
So these molecules are actually more granular as well as
Bigger in size.
bigger in size. Yes, size larger. ठीक है ना? From the plot you can identify, or database is there, they will give you this type of peak is for which molecule. Like in a mixture you have you want to measure all the WBC. Okay, in the blood sample you want to measure the WBC. So what will give you, you know na, WBC actually granulocytes bhi hota hai,
Mm,
agranulocytes are also there, huh? So whatever the more granular, they will give you S value more, whereas agranulocytes will give you less S value, right? So in this way, database is already there, or standards or references are there. From the scattering value, you can determine which which WBC is in higher concentration. Concentration of the WBC is also you can determine. Monocyte concentration is high or lymphocyte concentration is increased or not. Whatever the result from the interpretation, you can determine it. Okay. So if you consider here in this figure from this plot, you just see monocyte is there, some granulocytes is there because site scattering results more. Same whatever I have shown previously, same thing is given here. Nothing is. From the data interpretation, it is written that larger and more granulocyte cells are seen as a large population with a high SS and FS. So here you can also determine the number of cells for the granulocytes are more. You go to आज है ना कराटे क्लास ड्रेस पहने स्कैन करे दादा को बोलो पहना दे Okay, so what is happening? So here you can understand granulocyte size as well as concentration of this type of cells. It is higher than other cells. Okay, from the density of the dot peaks. है ना? Now, if you consider the monocyte, monocytes are large cells but not so granular. So Y value is less in case of monocyte. This is the monocyte. So Y value is less because these monocytes, I told you, monocyte means monocyte and lymphocyte, they are not granulocytes, right? So
Yes, ma'am.
for lymphocyte and monocyte, you will get less Y value. That means side scatter value. ठीक है ना? Now, smaller lymphocytes and lymphoblasts form a separate population. So lymphocyte size is smaller than monocyte. Okay. So lymphocytes value give you less forward angle light scatter. That means less X value is there because size is less. Lymphocyte size is less. So it is showing you uh, less X value. Okay. So in exam, they will give you this type of graph. You have to identify which molecule has the larger size, which molecule has the more number of cells. Okay. You have to determine from the graph. This is the main thing from flow cytometer. Okay. Now going to next part. दादा को बोलो पानी दे. Okay. Now, see, therefore, these cells can be separated into different populations based on their FS and SS alone. Okay, na? Now, in this case, FSCS is used to sort cells. So, in this process, we are sorting the cells, na? Separating all the cells. So, therefore, it is known as the FSCS. So, FSCS is used to sort cells and enrich for a subset of cells, which is often then studied in further details using flow cytometry or other analytical techniques. Now, flow cytometry is used for cell analysis and focused on measuring protein expression or co-expression within a mixed population of cells. Okay. Now going to application where it is utilized mainly. Many applications are there. Mainly we are analyzing the protein. Okay. So diagnostic process in the research and including simultaneous multi-parameter analysis of single cells, characterizing and defining different cell types and from the heterogeneous cell population. This is done. Now, if you consider other applications, so in that case, analysis of whole human blood, okay, analysis of human blood for diagnosing diseases. Now, sorting different blood cell fractions for ex vivo manipulations, immunophenotype analysis of murine blood to identify transgenic or knockout animals, sorting and analysis of slew of cell lines, characterization or isolation of rare cell types, adult stem cells, and cancer initiating cells. Everything can be identified. We are sorting actually cells here. So you can identify different cells. Now, multicolor flow cytometer. What is the main principle here? Previously, we have discussed that only one antibody was there, है ना? With one antibody, one type of labeling was there. Fluorophore were attached. But in case of multicolor flow cytometer, actually various antibodies you can add and various types of fluorescent compounds you can add. So simultaneously you can measure multiple samples. Okay, by measuring, so different fluorophores will give you different wavelength result. Okay, so you can compile together and identify various samples together. So that is the main thing. Here you can see ना, यहाँ पे देखो, one is the forward scatter. Now, side by scatter is actually involved for the one is side by scatter detector. This is there, but for the different fluorophore, you will get different fluorescent detector. Different fluorescent detector will give you different result. Okay, if three fluorescent compounds you have added, in that case you can get three results for fluorescent detector. Okay, so if two fluorescent compounds are present, then you will get two peaks. In this way, nothing else, everything same, but in this case you are adding multiple fluorescent markers. Okay, that is the thing. Our everything same thing is written here, whatever I told you. So clear what is difference between multicolor and single color? Enough.
Yes, ma'am.
Same thing is given. Yes. Now how it is. Now can you mute it whenever you want to talk? Just okay. Now measurement of scattering light and fluorescence. In this case, FS and SS light and fluorescence from stent cells are split into defined wavelengths. And whenever light is going towards the any substrate or particle, what will happen? It will give you different wavelength peaks. So split into defined wavelengths and channeled by a set of filters. As I told you, filters are given in the pathway. See here, different filters are there. These are all filters. Okay, these are filters after the scattering. So so all the scattered lights are channeled by a set of filters and mirrors within the flow cytometer towards sensors. Sensors, which is known as the PMT or photomultiplied tubes. So laser beam, then particle lights are scattered. Those are coming to filter, and after that there will be detection. Okay, so it is after that there will be detection, and that is through sensor, that is known as the PMT. So this is PMT. Okay, after filter PMT is attached. Now the PMTs convert the energy of a photon into an electrical signal. Now, up to this laser beam means it is the photon, light, photon particles. Now, photon is converted to into an electrical signal. Okay, and you will get voltage result. Now, fluorescent light is filtered so that each sensor will only detect fluorescence at a specific wavelength. So in this way, detection is done. Okay, FS detection, SS, and fluorescence detection is done in this way. Okay, now again I am showing you what are the things attached with the fluorometer. See, three systems: fluidic system, optic system, and electronic system. In fluidic system, two things are there. One is the core cell, that is flow cell, where sample is loaded. Second is sheath fluid cell, okay, where generally buffer saline samples are given. Okay, and now in the optic system, what is present? In the optic system, laser light beam source, then other filters, all the filters are involved, and then PMT, where sensor is present, right? Now PMT is attached with the electronic system. So PMT is actually converting photon to electric voltage. Okay. Now next part देखते हैं क्या है. Now what are the examples of fluorescent compound? Again it is given. In the example shown in figure 6, the fluorescent isothiocyanate, FITC. I have given example. FITC is the fluorescent compound. So the fluorescent isothiocyanate channel PMT will detect light emitted from FITC at a wavelength of approximately 519. So what I told you, in case of multicolor emission system, what is happening? In that case, multiple wavelength is involved for the multiple chromophores, no, fluorophores. है ना? So FITC is actually giving emitting light at 519. Okay. So in this, generally in the fluorescent, you are giving light, laser beam. If you put like laser beam, it will scatter in a different wavelength. Okay, you are scattering wavelength. We are measuring the scattering wavelength or emission wavelength. light of emission. So detect light emitted from the FITC at a wavelength of approximately 519. So emission light wavelength for FITC it is 519. But if you consider about the 5 phycoerythrin, this is also fluorophore. In that case, wavelength is 575. Therefore, only you will get different peaks for different fluorophores. From this picture, you can understand. See here, only two different fluorophores are given. If you consider one is for FITC, another is for PE. So you will get, see, this is PE. Orange color is for the PE. So you will get different peak. Now, if you consider this one, this is for the FITC emission. Now, other two emission is there, that is side by side scattering, and another one is forward scattering. So you get several data here. Okay, clear. Now cells stained with fluorescent antibodies pass by the laser, so you will get these multiple peaks, multiple result. Is it clear? Multicolor, है ना? So whatever we have discussed, that is actually we have discussed about the multicolor fluorocytometer. Okay, now going to next one, that is anything else is given here. Mm, here you have to select according to the availability and acha. Now cell populations can sometimes be separated based on if you select, but cells can also be separated by whether they express a specific protein. In this case, a fluorophore is usually used to stain the protein of interest. Okay, nothing else. Here only examples are given. What are the fluorophores utilized for this process? Okay. Now this staining is done by the fluorophore. So staining may be direct method or indirect method, exactly same whatever you understood about the ELISA and in case of western blot, here also same thing. Direct method means you are adding antibody, okay, and with antibody fluorophore is attached. Clear? So primary antibody with primary antibody fluorophore attached. That is the direct method. Now, if you consider indirect staining, in that case, you have to add the secondary antibody. And with secondary antibody, fluorophore is attached. See, conjugated secondary antibody, nothing else. So this is the detection purpose only. You can do with direct staining method or you can do indirect staining method. Clear? Hello?
Yes, ma'am.
So whatever we have discussed now, direct staining and indirect staining, those are actually coming under the surface staining. Cell surface, any receptor is present, you can do direct method and indirect method. Okay. So now another method, another thing is there, that is how you will stain intracellular compounds. एकदम स्टार्टिंग में मैंने बताया था ना कि टू टाइप्स ऑफ मॉलिक्यूल्स यू कैन आइडेंटिफाई, इंट्रासेलुलर एज वेल एज सरफेस मॉलिक्यूल, है ना? तो whatever we have discussed till now, we have discussed mainly about the outer cell antigen, okay, outer cell molecule, but you can detect intracellular molecule also, and that is through intracellular staining method. How it is done? In addition to cell surface antigens, the intracellular staining procedure allows direct measurement of antigens. Mainly in that case, intracellular molecules are cytokines, transcription factors, those are present inside the cell, right? So if you want to detect those protein molecules, cytokines or transcription factors, you have to follow intracellular staining. And in that case, mainly what is done, some permeability of the molecule or permeability of the cell is increased so that fluorophore can enter into the cell and can bind with these intramolecular antigens. Okay, so in this procedure, fixation and permeabilization of cells are required. Okay, fixation means fixing the antigen inside the cell at the single position. See here, it is written fixing cells retain the target protein in its original cellular location, fixing the cell, and will usually ensure better stability of soluble antigens and antigens with a short half-life. If short half-life is there, so you can fix it first and then increase the permeability, and finally you can add the antibody. Okay, labeled antibody. Same thing is given and nothing else. Okay, now sequentially it is written cell stimulation, example induced phosphorylation. Then fix cells, permeabilize, stain with fluorescent phosphospecific antibodies. Now you can analyze the sample through the fluorocytometer. Okay, and you will get multiple peaks for multicolored compound. Now data analysis, how we will do from the histogram. This type of plot you can get, like you can check the intensity of the light or emission light, and it is FITC at the right at the x-axis, and you can count the number of cells here at the y-axis. These rays are also possible. In that case, what will happen? This value is giving which result? In this case, y value is high, but x value is less, right? This peak, for this peak, first peak. That means FITC less, but number of cell at FITC condition, that means low emission is happening. In that case, size of the cell is low, smaller size, and smaller sized particles number higher. Okay, so y value is high. That means number of cells you can consider ki more, but size of that particle is less. Now, if you consider here x value is more, in that case, lots of events at high fluorescence intensity. So in this case, x value is high as well as y value is high. Okay. So from the graph, you can easily understand. Y value high means number of cell high, and FITC high means size of the particle high. You can consider. Okay. Now from this data already we have discussed. This is actually density plot, and in that case you have to measure forward angle light and side angle light, side scatter. From that you can identify which one is happening what. And this graph is easily understood. What do you think?
Yes, ma'am,
Anna?
understood. Mm.
This is very good. You can easily understand it. So there will be. So in this way you will get question paper, nothing else. So in this method, why you understood about it? Main thing in this case, we are analyzing cell, or you are sorting the cell types, है ना? And you can detect the antigen specific, and in this case main thing is that fluorescent compound is added. ठीक है ना? So this is new thing कि fluorescent compound only you can detect here. So flow cytometry test is usually based on measuring fluorescence intensity produced by fluorescently labeled antibodies. And in this case, actually, in the instrument, what is present? One cell is there. Which cell? बार बार भूल जाती हूँ मैं ये cell का नाम. Fluidic system. Fluidic system is there. एंड फ्लूइडिक सिस्टम में क्या करेंगे? सैंपल लोड करेंगे, और सैंपल जो है सिंगल सिंगल पार्टिकल के हिसाब से वो यहाँ से ड्रॉप के हिसाब से ये आएगा. ठीक है ना? कंटिन्यूअसली आता जाएगा, और लाइट बीम जो है, लेजर बीम जो है, it will be scattered into side light scatter as well as forward light scatter signal they will give you, as well as you have to measure the fluorescent emission light. Nothing else. Nothing else. A lot of things I have discussed, but those are not important. You have to just remember what is the structure of the instrument, okay, and what is the principle. Nothing else. Okay. I'll share one video so it you can understand Hindi, na?
हाँ मैम.
I'll share one video. That video is very good in Hindi, but it's very simply explained. I'll share that. Okay. Whatever I have discussed, everything is there. You can understand easily. Okay. And just a minute. Is there anything else is remaining? Just a minute. Hmm. Acha, in this case, these are the difference between FSC and flow cytometer. Normal FSC means FSC is used to sort cells and enrich for a subset of cells, which is often then studied in further details using flow cytometry or other analytical techniques. Flow cytometry is used for cell analysis and is focused on measuring protein expression or co-expression within a mixed population of cells. Acha. And that's it. Nothing else. ठीक है, so we are measuring mainly protein here, nothing else. Protein or cell, here specifically cell. A normal, if you just consider normal flow cytometry, you can consider ki protein, yeah, separating proteins. Okay, so now I hope it is done. Okay, so western blot, northern blot, southern blot, as well as we have done flow cytometer. Next class we will discuss fluorescence. Do you want to continue with fluorescent microscopy?
Yes ma'am.
Okay, then I can teach you, no problem. That is very short, short part. Achha. Hmm. Class flow cytometer blotting. Why it is not showing? Just a minute. Put out first. It is not showing. Blotting flow cytometer. Flow cytometer blotting techniques. Why it is not showing? I don't know. It is not showing. Don't save. Yes. Oh, yes, it is visible. I have it. It is visible, right? See, in this case, okay, the phenomenon of fluorescence was known by the middle of the 19th century, and British scientist Sir George Stokes first made the observation that mineral fluorspar exhibits fluorescence. The which mineral? Fluorspar. It is showing the fluorescence when illuminated with ultraviolet light. So whenever UV is given on fluorospar, in that case, what is happening? Some fluorescent light is coming. Okay, so fluorescence is coming. So when illuminated with the ultraviolet light, and he coined the word fluorescence. So this name is given by George Stokes. Okay, now in that case, what is the difference between the conventional microscope and fluorescent microscope? So we are talking about the fluorescent microscope. Okay. So fluorescent microscope means here fluorescent light is involved. Whereas if you consider the conventional microscope, in that case mainly we use visible light. So visible light wavelength is between 400 to 700 or 400 to 800 nanometer. Okay, so this is the wavelength. See, conventional microscope uses visible light, which is from 400 to 700 nanometers, to illuminate and produce a magnified image of a sample. This is the main thing happening in the conventional microscope. Whereas in case of fluorescent microscope, it is using a higher intensity light source which excites the fluorescent species in a sample of interest. Now, this fluorescent species in turn emits a lower energy light of a longer wavelength that produces the magnified image instead of the original light source. That means in that case, two types of wavelength would be there. One wavelength is which light you are giving, through which light you are exciting the molecule. And after that, what will happen? Fluorescent molecule will emit separate wavelength light. Okay. And after this emission, you will get image. So that is the difference. In case of visible light, you are just putting the light, and against that light, you are getting image. But in case of fluorescence meter, fluorescence microscope, generally light, whatever you are giving, source of the light will be different and emission light wavelength will be different. Okay, so you just read again. A fluorescence microscope uses a much higher intensity light source which excites a fluorescent species in a sample of interest. Now, this fluorescent species in turn emits a lower energy light. That means initially the wavelength of the light source is less. Okay, but after emission, the wavelength is higher. Wavelength is higher means energy is less. That you know, na? If wavelength is high, then energy will be less. So initially source of the light wavelength less, but energy was high. But whenever it is coming out of the fluorescent compound, the emission light is with lower energy with higher wavelength. Okay, now that produces the magnified image. So it will give you better results. So you can get uh, bigger sized image for very small particle. Okay, so which is not visible through the conventional microscope. You can detect it in fluorescent microscope because a smaller size particle can give you a magnified image in the fluorescent microscopy. Okay, clear? Is it clear? Hello.
Yes, ma'am.
So most in case of in this case, na source of light, this is visible light maybe or UV light, maybe UV light, because in initially he developed from he understood the topic fluorescence, that is from the UV light, na. See here with ultraviolet, whenever ultraviolet is first eliminated, this fluorosphere is illuminated with the ultraviolet light. That means source of the light is ultraviolet in that case. So ultraviolet plus visible both possible source of the light. But in case of conventional microscope, we are using only visible light. Okay. Now, what is fluorescence? Now, fluorescence is light produced by a substance when it is stimulated by another light. So this is happening now. Source of light is different, emission light is different. So fluorescent compound will give you different wavelength emission, light of the different wavelength. Okay, so fluorescence light is light produced by a substance when it is stimulated by another light. Without light, they will not give you any light, fluorescent light. Now, fluorescence is called cold light because it does not come from a hot source like an light bulb. So, fluorescent light is not coming from any light bulb, but cool. This is known as the cold light from the hot surface or hot compound. It is not coming. Okay, so it is known as the cold light. Now going to next. What is fluorescence microscopy? So in the fluorescence microscopy, whatever in this table is written, same thing here it is written, nothing else. See, fluorescence microscopy is a huh?
Ma'am, I think it will be better if we do the conventional microscopy first because, like, I don't know much about that one, microscopy.
Okay. But that topic is, you don't have to elaborately know this thing. Conventional microscopy, in order to understand the fluorescent microscope, you don't have to understand elaborately about the conventional. Okay, you understand what is happening, nothing basic concept. You don't have to know too much about this thing. Now, in bright field microscopy, specimens are illuminated from the outside, below, or above, and dark objects are seen against a light background. And in fluorescent microscopy, specimens are self-illuminated. So, in this case, whatever you want to analyze, any cell you want to analyze, na, in that cell, there should be some fluorescent compound. Fluorophore should be there. Then only you can get. So, specimens are self-illuminated here. Whereas in case of conventional, you are putting light on the specimen. Okay. Now, illuminated by internal light, so bright objects are seen in vivid color against a dark background. Okay. So you will get this type of image. On the dark background, you will get a very bright color sample. Okay. Now, if you consider about the normal light microscope, you will get normal, just shadow. This is white on the surface of white background. I think you will get the shadow of the same. Okay, in this way. In bright field microscopy, specimens are illuminated from outside, below, or above, and dark objects are seen against a light background. See, so on this background will be white, and cells you will get darker background. Not background, darker cells will be there. You will find dark part at the cell position, and surface will be there will be less color. Or you can see कि white background में black compound visible होगा. और whereas in case of fluorescent compound क्या हो रहा है कि on the black background you are getting colorful figure. ठीक है ना? That is the difference. Now, bright objects against dark backgrounds are more easily seen. Therefore, it is more favorable as compared to your conventional microscope. Okay, this characteristic of fluorescence microscopy makes it very sensitive and specific. Nothing else. Now, in this case, what is happening? Most cellular components are colorless. You know, now cannot be clearly distinguished under microscope. In this case, fluorescent dyes are given. So, fluorescent dye is also known as fluorophores or fluorochromes. Those are the molecules that absorb excitation light at a given wavelength, generally UV. Generally UV. So source of the light is generally UV, and after a short delay, emit light at a longer wavelength. So in that case, emission light wavelength is higher, energy less, and most of the cases you will get this light in the visible region. So wavelength will be like visible region. Okay, so I have given an example of chromophore in the fluorometry. In that case, 515 nanometer and 575 nanometer detection was done. है ना? So that means what? Same thing. उसका wavelength ज़्यादा है, in the visible region है. ठीक है, emission light का wavelength. Now the delay between absorption and emission is negligible. Generally, जैसे ही absorption हुआ, उसका emission भी immediately हो जाता है light का. Now, emission light can then be filtered from a excitation light to reveal the location of the fluorophores. This way, determination is done, nothing else. And this is the machine. You don't have to understand too much about this. Fluorescent microscopy uses a much higher intensity light to illuminate the sample. So in this case, UV light is given. See here, UV light is given. Now this is the exciter filter, and you want to measure this particle, not measure, you want to visualize this. So in that case, what is happening? Some emission light will be there. Okay, emission light will be there. So this is the fluorescent specimen, and you can detect it from here. Emitted blue light. Here in this case, UV light is coming, but emission light color is blue light. So this is visible light. So you can get blue color. You will see this molecule as blue color. Okay, na? So these fluorescent molecule will be blue color because it is emitting the blue color light. So detection will be for the blue color light. ठीक है ना. Now the image produced is based on the second light source or the emission wavelength. That is the thing. जो source of the light जो है that is UV, but emission light जो है that is visible light, so you will give color. So for the different fluorophore, different wavelength will be detected. Emission wavelength will be different, so you will get
Ma'am,
different
can you
color.
repeat? I lost connection
Okay.
like before.
Now see what is here happening here. In this figure, it is given what is the principle here. First of all, source of the light is the UV. I told you in last slide. So UV light is coming through the exciter filter. In the machine, it is there, and UV light is coming either UV or visible. I told you ना UV भी हो सकता है, visible भी हो सकता है, but most of the cases UV होता है. Now it is coming towards the fluorophore, जिसमें फ्लोरेसेंट कंपाउंड है, उसे हमें डिटेक्ट करना है या इमेज लेना है उसका. तो यहाँ क्या हुआ, लाइट इज कमिंग टुवर्ड्स दैट पार्टिकल एंड फ्रॉम दैट पार्टिकल light will be emitted. So if it is blue color blue color emission is happening, that means wavelength will be for the blue color. Okay, and emission light wavelength is blue color, and you will see color of this fluorophore is blue. Okay, so that means color of the particle is depending upon the emission wavelength of the wavelength of the light. है कि नहीं? So emission is actually coming from the fluorophore. It is emitting because of the presence of the fluorophore. It is emitting the light. Okay. And now, if the emission wavelength is for the green color, this compound will be like green color. Okay. If you consider that this is the green color fluorophore is given, that means it will emit the green color here. For different color, different wavelength is there. Okay, na? Visible light for different visible color, different wavelengths are there. Nothing. The image products is based on the second light. So image, whatever you are getting, that is mainly dependent upon the emitted light, not on the source of the light, whatever you are giving. Okay, emission wavelength of the fluorescent species rather than from the light originally used to illuminate and excite. So excitation is done by the source of the light. Every time I'm telling, okay, source of the light. That means starting point of the light, which light is going towards the fluorescent compound and exciting the fluorescent compound and illuminating the compound. Okay, by the emission of the light. Specifically, a dichroic mirror is used to separate the excitation and emission light paths. So यहाँ पे what is present, dichroic mirror, which is separating source of the light and emission of the light. Okay, within the objective, the excitation emission share the same optics. In a fluorescent microscope, the dichroic mirror separates the light paths. Now advantage क्या है? We same thing, nothing else. Fluorescent microscopy is the most popular method for studying the dynamic behavior exhibiting live cell. So if you want to identify bacteria by bacterial cell or any live cell, protozoa cell, you want to identify. So live cell, you can make the image of that through the fluorescent microscopy. So this stems from its ability to isolate individual proteins with a high degree of specificity, non-fluorescent. So if you can identify or you can separate fluorescent compound from non-fluorescent compound from this microscopic study. Now sensitivity is high enough to detect as few as 50 molecules per cubic meter. Very less concentration is there, then also you can measure. Very less size, smaller size is there, then you can also measure. Now different molecules can now be stained with different colors, allowing multiple types of molecule to be tracked. So if you want to identify multiple molecules, you have to add multiple chromophores. Sorry, again I am telling you chromophore, not chromophore, fluorophore. You have to add separate fluorophore. So separate fluorophore will label separate molecules, and from that emission light you can detect separate molecules. So these factors combine to give fluorescence microscopy a clear advantage over other optical imaging techniques for both in vitro and in vivo imaging. Okay, nothing else. यहाँ पे देखो, ये light is coming now, scattering, and there will be color. So this is emission light. देखो, this is source of light. It is coming towards the particle. From the
Can the
particle, it
video
is coming out of.
please
Again
start?
paused. Again
Yeah,
paused.
it's in slide number 8. I
Now slide
can't see
number
now.
10. Just 1 minute. Just wait for 1 minute. Maybe possible. You are facing so much of problem. I'm feeling very bad. Hello. Whenever you can be 10 slide is visible for you, then you inform me whenever it is visible. Slide number 10.
Looking, I'm still stuck in 8.
Now wait for 1 2 minutes. I'm waiting, no problem. You leave and again join if it is not happening. Okay. Now see what are the chromophores used. Whatever I have mentioned in my last lecture, na. So every same chromophores are utilized here. Sorry, fluorophores, fluorochromes. See acridine orange, auramine, slightly different, and fluorescent antibody are the fluorochromes mostly used. So these are the examples. What are the fluorochromes are used? Mainly auramine O and fluorescent antibody. Now, how to use a fluorescence microscopy? Now, this see the object to be how to use fluorescent. Okay, the object to be studied marked with a molecule called a fluorophore. So whatever you want to identify, if you want to identify any cell, you have to first of all you have to add fluorophore with that cell. You have to tag the sample with fluorophore. Okay, so the object to be studied is marked with a molecule called a fluorophore. Now fluorescent light is activated, illumination is separated. Now you can get excitation filter is given plus emission light. From the wavelength you can detect. Okay, and from the how much transmittance is plotted in the result. Okay, in the database you have to plot transmissions versus wavelength. Okay, so initially you will get exciting filter wavelength, and finally you will get emission filter by wavelength. Okay, and intense from the intensity, that means this area calculation that is automatically done in the lab computer. Okay, and from the intensity you can identify how much amount of fluorophore tagged cell present or not. Okay, so that is the thing, nothing else. So we are plotting here transmittance versus wavelength. Okay, in this wavelength your exciting wavelength is also given and emission wavelength also given. Okay, now why it is so badly given? Look at the light. So what are the steps involved in this process? Locate the light switch on the side of the microscope that turns on the light. So you have to switch on all the machine parts and write down the exact time you turn on the light. Now the Fluorescent light is mercury based, and a time log must be kept for exposure and use the light. So you have to measure the time when you are switching on. Now, second step is locate the toggle size switch on the right side of the microscope between the oculars and objectives. This switch controls the shutter for the mercury light to the objective lens. Hey, nothing else. Yeah, you don't have to understand these things. You just on all the switch. Now, step three is select the appropriate dye. Okay, now this is you can consider the sample preparation. Select the appropriate dye for your object. The most common dyes include, these are the different dyes. 13 for use city. These are the name of the compound. If you possible, you remember it. And the PF420 N21 L3. These are the chlorophores are utilized during the detection of the cell. Okay, different slide, different number of chlorophores are given. Now put the filter. So next step is put the filter into the tray operated by the silver sliding knob. Now remove the tray, simply pull the silver knob out. How it is possible to understand in this way? It's not possible to understand. Okay. So now, in this case, it's like a normal microscope. We generally check in 10x, 20x, then 100x resolution. So, in this machine also, this type of lenses are there. You can watch through 63x and you can watch through 100x. Okay, and you just now turn the light off when finish time. Wait 30 minutes before turning the light back on or lamp could explode. It is a good idea to keep track of how many hours the lamp is used. Replace according to manufacturer's guideline. These are nothing else. Nothing. Now clean out the microscope lens. Say these are the basic things. Nothing. You don't have to understand what are the methods are given here. Okay, so very stupid things are given, nothing else. If I find any video, demo video, I'll share that. From that you can understand properly what is the machine is. Without watching the machine, it is very difficult to understand function of the machine. Okay, so this basic principle you just understand key. One wavelength will be for exciting light and another wavelength will be for the emission light, and emission light wavelength is higher than exciting light wavelength, and in this way we generally detect the emission light. You can find the different color for different fluorophore. Okay, so in this process you can measure small particles. You can detect small particles, small molecules. Live cells can be stained. Again, live cell can be detected through this type of microscope. Okay, nothing else. Don't know what type of questions is coming from this topic. I have to check that. If I find any previous year question paper related to this microscope, I will share that. Okay,
Okay, ma'am.
you just understand basic what is the principle, nothing else. Everything is not required. Chalo. For today, I'm leaving.
Okay, ma'am. Thank you, ma'am.
Okay. चलो ठीक है. And simultaneously, are you studying my PPT or not? After the
Studying, ma'am.
class, just go through once. Just go through it, and whatever I'm sending you the question paper, you solve it. Question paper means just video I'm sending. Na? You just check those.
Okay, ma'am.
Okay. Chalo. Bye.